So for those of you who don't know me, I'm Lauren Toth. I work over at USGS studying coral reefs. And I'm very pleased to be hosting Dr. Ian Enochs today. He is the head of the coral program at NOAA's Atlantic Oceanographic and Meteorological Laboratory in Miami, Florida. He's a research ecologist there. He started his career at University of Miami, where we actually met in undergrad, and got his PhD at the Rosenstiel School of Marine and Atmospheric Sciences, also in Miami. And with that, I'll turn it over to Ian to tell us all about his research. Thanks, Ian.
I appreciate that, and I hope people online can hear me. Thank you guys for having me. It's a privilege to be here and a privilege to be able to speak about some of our research. My research portfolio expands to a bunch of things that I will not be talking about today, including some collaborations with some of you here. I'm happy to talk more about coral disease, more about eutrophication, more about restoration later on, over a beer preferably. But today we're going to be really focusing on the persistence of reef habitat in an era of global change. And before I do that, because I know not all of us are reef-centric, I want to take a second to really think about why we care about coral reefs. I think this is important. If we're going to spend 40 minutes talking about coral reefs, let's think about why first. So first of all, and as many of you well know, they exist as the highest concentration of metazoan biodiversity in the marine realm. They are the rainforests of the sea. Part of this biodiversity, fish, are incredibly important for coastal communities, for putting protein and food on the table to countless people. In addition, as several people in this room have shown with their research, coral reefs are a living wall that provide protection to our shorelines, to our coastal infrastructure, billions of dollars of protection from storm surges and wave energy on a yearly basis. They also provide billions of dollars of economic stimulus through tourism and through diving and snorkeling. And all of these billions of dollars are why I likely have a job in NOAA in the US government, which is under the Department of Commerce. But I also want to be really thoughtful about the fact that none of these anthropocentric services are why I started studying coral reefs. As a small child in the Midwest, I was drawn to that sense of wonder, that sense of discovery, that sense of adventure. And I think, as scientists, we should not keep that out of our talks. I think that's an important, especially as we're communicating to wider audiences, to keep that up front and center when we're talking about these ecosystems that we love and that are imperiled. But the problem with that is that I can't really quantify that aspect. These other things, however, these other ecosystem services, I can directly tie to one specific aspect of a coral reef, and that's habitat. And this habitat is formed by the precipitation of calcium carbonate, the calcification, by primarily scleractinian corals. But what you likely have not seen, if you're simply diving on a coral reef, is this complex network of cavities, of boreholes, of cracks and crevices that are formed inside these skeletons by a biodiverse suite, a multiphyletic suite of organisms collectively termed bioeroders. This is, for all intents and purposes, the antithesis of calcification. And I want to dive into this for just a second more, because it will become important later on in my talk. But these bioeroders can be divided further into microborers, which, as the name implies, are microscopic. They use specifically chemical dissolution to dissolve away those carbonates. There are also macroborers, including clionid sponges, lithophilic bivalves, various annelids that use a combination of chemical erosion as well as mechanical abrasion. And the organisms that you are likely most familiar with on the surfaces of a reef are the grazers. These are parrotfish, these are urchins that are using mechanical means, mechanical abrasion to snip away, rasp away, scrape away bits of that calcium carbonate. And again, on the other side of the scale, we have calcifiers, primarily scleractinian corals on the reef. Though crustose coral and algae is very important for this. And so I'm trying to be, play to my audience. But anyway, so we have this incredibly complex subtraction equation right here, right? Where reef habitat over time is simply equal to calcification minus bioerosion. And we can take this a little bit further and add this up for all of the species on a coral reef that are involved with these various processes and multiply it out by their prevalence. And we can do this in my backyard, in our backyard, in the Florida Keys. And it gets a little bit more complex because we have a lot of species involved. And we also have a lot of species involved on the bioeroding side. Different size classes of parrotfish, different life history stages or life stages of parrotfishes. And all of these things matter and make that simplified equation just a wee bit more complex. But we've done this, by that I mean one of my graduate students has done this in the Florida Keys. And you don't need to be a coral reef ecologist to see that there's just straight up a lot of red on this graph, right? And that is because a lot of our reef, I believe more than 70 or 80% of our reef in the Florida Keys is actively eroding. And these colors, we're looking at, roughly, in the ballpark with this red here, around 2 kilograms of calcium carbonate per meter squared per year. And I want you to take a second to think about in your hand what 2 kilograms of calcium carbonate feels like. And then I want you to rip that, not in the real life, but in your minds, away from a meter square of reef every year over decades, over multiple decades, and think about what that actually means, what these simple little numbers mean for the erosion of that habitat and all of those ecosystem services that we were talking about that are so vitally important for our economies and our communities, what that actually means. And it's a lot. A meter squared, a kilogram, a couple kilograms every single year. But this graph is also important. We're not going to be strictly negative through this talk. We're going to be mostly negative, but not entirely. There are still some reefs that I want to point out that are still well in this green zone, particularly here in this mid-channel environment. And that's important to keep in mind because all is not lost, and there are still some areas that are actively growing. But this is today, this is right now, and we all know, and I hope this is not a shock to any of you, that our world is changing around us. Again, I hope this is also not a shock, but humans are creating a lot of carbon dioxide. It's warming things up, and much of that carbon dioxide is also absorbed by the ocean, where it combines with water and ultimately produces hydrogen ions, lowering the pH in a process aptly termed ocean acidification. And we know for a fact that this is happening. We know that atmospheric CO2 is rising. This is not just some hand-wavy science thing. We know that atmospheric CO2 is rising because we've measured it. We have also, not for the same amount of time, but measured a very significant increase in seawater CO2. And this has been accompanied by a decline in seawater pH. Again, ocean acidification. This is not something that is happening only in the future. This is happening right now. We know this process is real. But it will continue to happen in the future. Climate modeling has shown under various emission scenarios that if we don't severely change our behavior, we can expect a drop in seawater pH of roughly 0.2 pH units by the end of the century, depending on our behavior and what we do. And for those of you that don't think the 0.2 is a lot, consider that pH is obviously on a logarithmic scale, and that this could have very profound ramifications for the physiology of all the organisms that live in the sea. But things are not that simple. Carbonate chemistry and ocean acidification conditions vary in both space and in time. And this is especially true in nearshore environments. And if we go back to the Florida Keys, we have, in collaboration with some of you that are in this room, we have been conducting surveys of carbonate chemistry, taking water bottles every couple of months across these transects across the Keys. And we've been analyzing this for a couple different carbonate chemistry flavors. We've been looking at dissolved inorganic carbon as well as total alkalinity. We do a bunch of spectrophotometric pH, but I believe in these plots we've solved for pH in aragonite saturation state. Aragonite is the crystalline form of calcium carbonate that corals precipitate, is especially relevant to coral reef research. And what you can see here is months across the x-axis. We have, and across these different columns, we have different regions in going off the shelf in the Florida Keys. And it's pretty clear. In these inshore environments, we have these strong seasonal fluctuations. This makes a lot of sense because we have changes in productivity over these seasons that ultimately drive the carbonate chemistry around it. Changes in photosynthesis and respiration that ultimately lead to these strong seasonal oscillations. And this becomes less pronounced over time as we move steadily outward to the oceanic. So strong benthic forcing in these shallow environments. And this matters for a variety of different reasons. This is our trend detection of ocean acidification over time in these same different regions. And this is, again, some of the different carbonate chemistry parameters. What you can see here is, as we move offshore, we are able to detect a clear ocean acidification symbol, but in these inshore environments, this variability obscures that detection. Also important in this is that you see that these organisms are routinely experiencing incredibly high CO2. Ocean acidification conditions that are projected in some situations to occur roughly near the end of the century, already experiencing it every single day or seasonally. So we know it's a mess. We know that these things are happening. We've got this complexity in space and time. So the question then becomes, how do we actually understand, how do we study, how do we predict the future of coral reefs today? Well, one of the approaches that I have taken is to build experimental facilities that allow us to manipulate carbonate chemistry in real time. This is the experimental reef lab that I have at the Cooperative Institute for Marine and Atmospheric Studies at the University of Miami. It's joint with AOML. We are constantly running a lot of experiments in there. We've designed our own hardware and software to control these. Everything fluctuates. We're able to program everything in on hourly dependent set points. And I say this because most everyone, a lot of people boil and pickle marine organisms to see what will happen. But what we can do is we can change those fluctuations. We can change the magnitude of those fluctuations. And this is important because there are, in fact, diel fluctuations, just like the seasonal fluctuations we're talking about. There are diel fluctuations. Obviously, calcification and dissolution are light mediated, just as photosynthesis is. Makes total sense. And so here you can see our ramping of conditions, or I hope you can see ramping of conditions with a mean pH. Roughly, these colors will stay the same in the next couple plots. Mean pH, contemporary pH, a future pH ramping, and then we have 3 diurnal fluctuation scenarios, no fluctuations to high magnitude oscillations in the future, and two in contemporary. And so that's not enough. We also threw some cropper cervicornis in. And we found that there was significantly enhanced calcification in contemporary conditions. So not just the mean, but rather the magnitude of that oscillation is structuring the response. Now, as should come to no surprise to many of you, there are also significantly strong differences in growth performance of different genetic individuals. And so if we impact this a little bit more, we can actually see that there's this general trend in these future conditions of a reduction in growth and higher magnitude oscillations, whereas in contemporary conditions, it's quite the opposite. So again, there's this switch. It's not just your mean pH value, but it's that fluctuation that you're experiencing in a day-to-day cycle. There's also this little friend here that is not responding at all, couldn't care less. And that is really important as we're starting to do managed or informed climate resilient restoration. We need to be identifying these individuals that are not particularly responsive to these stressors. We also did this, we have done this on several different bioeroders. This is a species of clyonid, 2 species of clyonid sponges that are very intense bioeroders on coral reefs. And again, we found that there are strong differences based on not just that mean, but also the magnitude of the oscillation. Starting to layer on this complexity of what we're looking at. But I will be the first to mention that every single experiment that I've run in that lab has been inherently flawed. Not wrong, but inherently flawed. And this is because we cannot fully program in all sorts of oscillations. We can't fully capture that wild variability that we were seeing in those plots earlier. Also, experiments are expensive and are really time intensive. And so they're often limited in duration. And so we can, we can miss more subtle effects, as you can see here in light green, and also we can misinterpret things that would be potentially acclimatizing if we looked at scales over time. Most of our experiments only look at a single species, and most of those only encounter a single stressor. But we know in the real world that we're dealing with a myriad of different stressors simultaneously impacting these species, and that these species are simultaneously influencing each other, competing for space, competing for food, preying upon each other. And so what we actually have in the real world are levels of complexity that are orders of magnitude higher than we can expect currently to be able to replicate in the lab and ultimately understand so that we can better predict and manage these ecosystems. So as I have struggled with, how do we incorporate this environmental complexity with this ecological complexity, and how do we do this over longer periods of time? And one of my approaches to dealing with this is to have some wild adventures. This is in the Marianas Islands. This is the island of Mog in the far northern Marianas Islands, and you kind of travel up all these deserted, kind of smoldering volcanoes. It's absolutely beautiful place. Took me several years to realize that Mog is Guam spelled backwards. So it was like, seriously, I've been studying this place for a while, and then I was like, oh, wait a second. Yep. Anyway, so once you get to this site, it's pretty, and I say this word strongly. It is a pretty magical place. It is a calm, kind of almost like a lake in the middle of the ocean, right? Like it is protected on all sides. You're lucky to be here, and full moon in this area sampling. It is one of the most amazing places that I've ever been. That doesn't matter for this talk. What does matter is that if you dive under the water there on the inner margin of one of these islands, there's this area where there is active venting of subterranean carbon dioxide gas trapped, because of the volcano, up through the seafloor. It is like diving in champagne. It's absolutely wild. And what it does is it creates this localized ocean acidification hotspot. Doesn't matter on a global CO2 cycle, humans have that covered. This is something different. It is a very small, localized experiment that has been going on for quite some time, involving numerous species constantly interacting. And so we first set out to characterize the acidification gradients at this site so that we knew what we were dealing with. We picked high and low and mid CO2 sites, as well as a control that's slightly off this map. And we specifically targeted them to be in conditions that are realistic. We didn't want to be up in this higher area where it's analogous to if we burned every bit of carbon on our Earth and maybe a little more. But we instead picked conditions that are typifying these mid and end of the century scenarios. Also looked at their fluctuations over time. So again, we're trying to capture both spatial and temporal variability. And the question then becomes, how are these acidification gradients? Great, we have this natural laboratory. What's this doing to these ecosystems? Well, one of the approaches that we use are structure from motion and mosaics. So you guys are probably familiar with these. These are roughly 10 by 10 meter plots. They still have such high resolution that you can zoom in and look at little polyps and identify everybody and figure out what's going on. But this is my favorite kind of science because you don't have to be an expert to really see it, to really see what's going on here. We go from this situation with contemporary CO2 as we start to ramp up these ocean acidification conditions, to a situation where we're dominated by macroalgae. And I should say that these boulders are not happy coral reef. It's just a bunch of algae growing on rocks, and it is not a fun place to dive unless you're nerding out about these kind of things. And I should say that it was a pretty interesting thing to do because I had been studying ocean acidification in tanks for several years, and I never really felt it, never really experienced it until I jumped in here, and it was a very transformative, very important moment for me. But because we're scientists and we don't just look at pretty pictures, we also quantified this, and we indeed, you can see these classically predicted, coral goes down with high CO2, algae goes up because it photosynthesizes, and that makes total sense. But where things start to get a little bit interesting is when we start to look at turf algae and crustose coralline algae, we have higher abundances in this intermediate CO2 environment. So experiments, excellent experiments by people in this room, have shown that some of these should be responding quite, linearly. We expect crustose coralline algae to be going down in high CO2 conditions and turf algae to be going up. So what's going on here? I don't know. But I expect that it could have to do with competition, and that the presence of all of these different competing functional groups could be that in these environments, for instance, maybe coral or maybe fleshy macroalgae is reducing these other functional groups and these other taxa to a point where we have these somewhat unforeseen ecosystem responses. And so we wanted to dive into this a little bit more and figure out what biologically drives this. We, again, like many people in this room, we employ coral coring, where we collect a drill underwater. This is Galapagos, it's not there, but it gets the picture, it gets the point across. We take these back to my lab where I have a CT scanner, and we CT scan these and ultimately arrive with a something like this. And corals put down these yearly growth bands, just like tree rings. And we can trace a growth axis over time as it kind of zigs and zags along as it decides where it's wanting to grow over time. It can be really annoying when they move out of your core. But, and so we can understand how calcification and how linear extension has changed over time, get a good understanding of how stressors have been impacting it, compare different areas. And that's what we did here. We found reduced calcification in this more acidified environment. Now, we also wanted to look at the other side of the scale. We wanted to look at bioeroders, but we were really limited in the amount of time that we could be at this site. So we decided to focus on microborers that colonize quite quickly. And we used Icelandic spar. And while this looks like some sort of healing crystal or whatever, that's where we got it, but it is crystalline aragonite. And so it is the same mineral structure of a coral. And we threw these out for 3 months. And again, this is quite intuitive science. You can see in this high CO2 environment, much higher colonization of these borers, these microborers, than in the low CO2 environment. And I should point out that these pictures are not just on the surface of these little fragments. This is because, these are transparent. This is these boreholes that have been created inside of them. So pretty interesting. But there is to butcher the quote of "No man is an island." As we've been discussing, no organism is an island, and the question then becomes: do these patterns manifest if we are looking at multiple functional groups actively competing? And to do this, we're going to hop on over to another adventure in Papua New Guinea, where I've been working with the Australian Institute of Marine Science and Katarina Fabricius's group. And we create these extremely complex, and by that I mean not at all, chunks, domino-sized chunks of calcium carbonate. These are Porites lobata skeletons that we simply glue to a piece of PVC, and we simply chuck out on the reef and go back and do our thing. This is next to a CFET pH logger. And so in Papua, we did this across a couple other high CO2 events. You can see these bubbles bubbling on over here. And we waited. And over time, things grew on them, things grew within them, things broke away parts of them, and they became, in effect, little chunks of the reef, which we unceremoniously pulled off of the reef and brought back to the lab. And here you can see that we have these from different, ocean acidification, mean ocean acidification scenarios. I also have a micro CT, and this is essentially a CT with the word micro in front of it, meaning it's higher resolution. And this allowed us to volumetrically isolate the different individuals operating and growing and ultimately perturbing the calcium carbonate on each of these little dominoes over several years under different ocean acidification scenarios. And this looks like this. Chunk of skeleton. You can see the crustose coral and algae growing on the outside of it. If you take the skeleton away, you can see this complex network of boreholes of here annelids that have eroded away the interior of this little chunk of reef. And I told you we'd be talking more about those functional groups. And we are, so we were able to quantify each of them in various different ways: grazing, macroboring, accretion, and microboring. And we were able to look at this across those ocean acidification scenarios. And these plots are a little bit awkward because low pH or higher ocean acidification conditions are on the left, temporaries on the right. But you can see indeed that there is strong enhancement of bioerosion in these more acidic conditions. And it is more pronounced than that of accretion, which is a little bit of my soapbox issue, but what people mostly look at with respect to ocean acidification. This was the first time that anyone has shown that annelids, worms, were accelerating a bioerosion under ocean acidification conditions. Yeah. We also looked at grazing and microboring, as I said. And was not a surprise to us that grazing was not significantly impacted by ocean acidification because fish swim in and out. These are not huge ocean acidification sites, so they're swimming out. They can move in and out as they well please. But what was surprising from our work, from others, is that we didn't detect a significant trend in microboring. I don't know why, but I have some guesses. I do think that it is possible when we're starting to look at substrates where we have grazing, where we have the removal of those upper substrates, where these fish are potentially even getting nutrients from grazing on these endolithic microbores, that it obscures that signal, that ocean acidification enhancement of microboring. And so in these scenarios, it may not be as ecologically relevant. But that being said, many people fish parrotfish. And so if we remove all our parrotfish, perhaps this contribution will be more important, particularly in the future with ocean acidification scenarios. We also conducted alkalinity anomaly incubations on these. These are similar to if you've done respirometry. You just take a fixed chunk of water, you chuck your organisms in it. You know the carbonate chemistry. The total alkalinity is only influenced by the balance between calcification and dissolution by erosion. And we did this to these communities in the light and in the dark and averaged it over 24 hours. And we saw this tipping point around a pH of 7.8, where these little bits of reef go from net growth to net erosion around 7.8. And those of you that were paying attention early on probably saw that 7.8 is around an end of the century condition, which should be scary to you. But this is also overly optimistic because it doesn't include grazing. It doesn't include mechanical erosion. And so in all actuality, I expect this tipping point to occur at much more conservative ocean acidification scenarios than the end of the century in this condition. I'm going to hop on over to another place that I've been lucky enough to work with Dr. Toth on. This is the Pacific side of Panama. We have the Gulf of Cherokee and the Gulf of Panama. And one of these areas, the Gulf of Panama, is strongly upwelling. The other Gulf of Cherokee is mildly upwelling. And this means that we have higher acidification conditions in the Gulf of Panama, these CO2-rich deep waters, but higher productivity, also a little bit cooler. So these two very different environments. And don't let the shape fool you. This is the exact same thing that we did in Papua. And we essentially just unceremoniously chucked a bunch of reef rock out on the reef and then waited for a long period of time. And this time we quantified communities, not just in terms of the overall functional group, but we delved a little bit further into their taxonomy, using their traces to identify who they were and what they're doing. And we did this in both Gulfs. None of that's really important. But what is really interesting to me is in the Gulf of Panama, higher CO2, higher productivity environment, we measured higher calcification and higher macro boring, which is kind of contrary to what you would at first pass think. What's going on? But when we looked at what was actually causing this, we could see that these are primarily heterotrophic taxa that are probably responding most closely to the higher productivity. These are vermiform and barnacles, suspension feeding taxa. And these guys are creating carbonates, but they're creating carbonates that are not as stable and are not as, if I may, valuable as those created by corals, because they are, eroded over time and more likely to turn into sediments. The higher macro boring was also another heterotrophic species, lithophage bivalves. And so I mention this because you can't just assume that this is ocean acidification, even though we have a strong ocean acidification scenario. There are other stressors that are involved, and depending on the unique physiology of the constituent taxa that you're dealing with, something entirely different might be happening. Where we're moving with right now, this is a paper that's in revision, about to send this back, is we're adding further complexity, going down the complexity rabbit hole of starting to deal with natural reef substrates. We're starting to deal with rubble, subjecting these to different ocean acidification conditions. Rubble is a pain in the butt to deal with. It is all sorts of interacting species simultaneously, all sorts of different sizes, all sorts of different densities. We go from our very consistent little bits of reef to wild levels of complexity. We're starting to be able to manage it and deal with it. Speaking of management, why are we doing all of this? Well, one of the examples of why I care about all of these responses, and one of the ways that we at NOAA are starting to look at this with respect to overall management, is something that we're doing. We've done at Chica Rocks. We're now starting to expand to the other mission iconic reef sites. We're looking at it throughout a lot of US jurisdictions. As we're creating these dynamic carbonate budget models, where we are able to parameterize the abundances of all of these different things operating on that habitat that provides our ecosystem services. But we are factoring in the various global change and stressor conditions that alter their action. And we've done this in different climate scenarios, looking at the overall ability to keep up with sea level rise, to produce height, to produce calcium carbonate. And we have done this under various restoration scenarios and also under various adaptation or acclimatization scenarios. Take home here, I'm not going to spend a lot of time on this, is it really takes everything, all hands on board. We need to be actively dealing with restoration, we need to be actively mitigating climate, and we need to be actively enhancing or encouraging organisms' ability to adapt to global change. If we hope for these sites to be continuing to grow and to persist and create habitat over time. So we're not in a great situation, but there are still ways to get out of it. So I also want to kind of pivot here. I want to talk about something else. I think that there's potentially a lot of opportunity for collaboration. I am the climate PI for the National Coral Reef Monitoring Program in the Caribbean and Atlantic. I also do a bunch of this stuff in the Pacific. But we're actively trying to characterize the progression of ocean acidification and warming on US coral reefs. And we're also monitoring the impacts on the important habitat altering flora and fauna that are at these sites. We are rapidly developing technologies and they're really excited about a lot of structure from motion stuff, starting to do this in 3D and look at overall changes in structure. We're also doing traditional carbonate budget analysis. And we ultimately produce these reports, which, it's like you're gonna throw a little grade on an entire reef ecosystem. It's like, part of me, it's hard to do, but it translates well to communities that really genuinely care about the health of their reefs and the severity of these different climate impacts. So we've had a lot of success in bringing these reports to various entities, management entities, so that we can distill down, not just me and my climate stuff, but fish and benthic communities and socioeconomic aspects as well. And it's really a holistic kind of approach that I'm very proud to be part of. So, we talked about complexity a little bit, and it strikes me that this talk could also be named, like, down the rabbit hole of complexity and towards insanity. But I want to really underscore that if we really want to be serious about predicting and managing these coral reefs, we really need to be dealing with complexity. And those of you that are involved with marine experimentation, I think many of you here are, how many stressors have you looked at simultaneously? Maybe one or two, three tops? And of those, how many individual levels have you applied to your organisms of choice, right? Have you high, mid, low, or is it a continuum? Because nothing in the real world operates on a high, mid, low basis. We're not categorically stressing things out. It's all on a continuum. But the problem with that is as we start to incorporate these layers of complexity, our experiments get complex to the point of unmanageable, financially unmanageable, logistically unmanageable. And to deal with this, people have created various statistical designs to identify combinations of stressors that they think are especially relevant, especially poignant to directing and altering the fate of an ecosystem. But one of the reasons that I am a scientist is because I like things that are unknown. I like to find something new. And it strikes me that this eliminates some of that. There are synergistic, unexpected synergistic effects of these multiple stressors. There are unexpected antagonistic effects of these multiple stressors, and there's unexpected stuff. And so again, if we were really looking to parameterize these models that will ultimately guide how we make management decisions, we got to know that stuff. And so I'd like to suggest that there is another way now that is perhaps facilitated by some of the technologies that are only now becoming more relevant, that we can go from these single individuals in these little aquarium, maybe dealing with 16 different aquarium treatments, that we can go to these complex designs. And so this is a system I published a couple of weeks ago. This is called the STAR system. It's a, closed by that screen, but it's the sequential treatment application robot. And what this does is it sequentially interrupts treatment application. There's a robotic arm that day in and day out applies different treatments. We'll inject, for instance, we've used this with diseased water. We use this at different levels of nutrients. We are using this with all sorts of different things. That day in and day out, to every single one of these beakers in arrays, we'll inject precision quantities of these various stressors. We can do this not just in combinations, but also in continuums of stressors, not looking at high, mid, low CO2, but rather 256 levels of pH. Start looking at regression designs rather than ANOVA designs that are, again, much more relevant to ocean acidification or whatever type of management model that you want to build. This is not just a CAD pipe dream of mine. This is something that is operational in my lab right now. We have these, this is, I believe, Armanda. We also have Armand. And also Dr. Mechanical Studivarm, which is another guy in my lab. But we've been successful at maintaining corals for periods of months, subjecting them to these combinations of different stressors. Again, expanding out our ability to do this type of research. This was not possible years ago, but with the advent of these robotics and the advent of, I think, financially reasonable, feasible robotics, this is now possible. I should say that the paper that I published on this, I've made absolutely everything open source. All the hardware I designed, all the CAD I designed, all the software, everything is open source, and we have directions for how to build this yourself. Because again, we want this to be available and modifiable and not self-controlled. So if you want to build one, I have directions. And they're on the web. So another direction that we're going in right now, Frank and several others of you are involved with this, is we're just lucky enough to be funded by NCOS for a large multiple stressor grant. This is a multiple institution consortium. This lab that I had showed you, we're now in a state of chaos because we have torn down all of our facilities and are building up again while still trying to maintain multiple experiments. And I'm kind of stressing out about this right now. But Project Fresca will ultimately allow us to look at a whole suite of different stressors impacting very important ecosystems in our region. All of this is based in the Florida Keys and around in Florida Bay, so South Florida region. And it incorporates very important, arguably, I think in the US, some of the most relevant restoration and management scenarios now. So we're looking at various Everglades restoration scenarios, as well as the most important coral reef restoration project, Mission Iconic Reefs right now, as well as various climate scenarios. Again, trying to look at all of these different stressors to see how they impact these ecosystems so we can create these tools and evaluate them is a massive, massive undertaking and something I'm incredibly excited and optimistic about. With that, I'd like to thank you guys and my lab and my funding sources, and yeah, I think I'm running 40 minutes, so cheers.